హాయ్ ఫోక్స్, వెల్కమ్ టు మై ఛానల్ ది యావరేజ్ డర్మాడాక్. ఇక మొదలెడదామా? Biology of mycobacterium leprae. This mycobacterium leprae cannot be grown in vitro. It cannot be grown in any lab because there is no culture media, no bacteriological media and no tissue culture to grow. It is an obligate intracellular organism showing tropism towards macrophages and squamous cells. Tropism means having a selective preference towards any cell or an organ. Here our leprabacilli have tropism towards macrophages and squamous cell inside human body. It is the only pathogen affecting superficial peripheral nerve. Only human pathogen capable of invading superficial peripheral nerve. It has a preference for temperatures less than 37 degrees. Hence grows well in cooler parts of the bodies like skin, testis, peripheral nerves and anterior part of the eye. It is an acid-fast, gram-positive, capsulated, non-motile and non-sporing organism having microaerophilic with shape straight rod or slightly curved rod shaped. It may be present as single or it tends to form clumps called globi because of the presence of lipid-like substances known as glia. In globi, if they are arranged in parallel row, it gives an appearance of cigar bundle. On electron microscopy, structure of mycobacterium leprae. We will see from outside to inside. Outermost structure, capsule. Next, cell wall. Next, cell membrane. And then innermost structure, cytoplasm. First, we will see about capsule. Capsule contains mainly bacterial lipids, phosphatidyl inositol monocytes, phospholipids, PDM, thiocerol dimycoserosate, PGL1, phenolic glycolipid 1. Mainly you have to remember PDM and PGL, thiocerol dimycoserosate, phenolic glycolipid 1. Next, coming to next structure, below capsule, cell wall. It is divided into electron transparent zone and electron dense zone. Again, electron transparent zone is divided into outer leaflet and inner leaflet because it is a pseudo lipid bilayer. Outer leaflet contains again same PDM and PGL1 which is present in the capsule extends to cell wall also, PDM, PGL1. Only one extra thing we have to remember is TMM, trehalose monomycolate. Coming to inner leaflet, it contains mycolic acid and arabinine chains. Coming to electron dense zone, again arabinine, galactan, lipomannan and peptidoglycan. We will see in detail about cell wall. Cell wall gives definite shape to the bacilli and protects bacteria from environment. Already I told you cell wall is divided into two layers. Outer electron transparent zone, inner electron dense zone. Coming to electron transparent zone, it is a pseudo lipid bilayer again divided into outer leaflet and inner leaflet. Outer leaflet contains mycolic acids of trehalose monomycolate. Only one thing extra was TMM, trehalose monomycolate. Remaining two things same as capsule, thiocerol dimycoserosate, phenolic glycolipids. Mycoserosic acids of thiocerol dimycoserosate, PHTHI, thio. In phenolic glycolipids, the dominant lipid in cell wall which gives immunological specificity was phenolic glycolipid 1. This interacts with laminin of squamous cells. Thus, it attacks peripheral nerves of human body. This bacilli also secretes this phenolic glycolipid 1 locally within the macrophages, giving it a characteristic foamy appearance. This foamy appearance having macrophage is known as Virchow's lepra cell or foamy macrophage, mainly seen in lepromatous leprosy patients, having more number of bacilli inside the macrophage, producing more amounts of phenolic glycolipid 1, giving it ultimately foamy appearance. Now coming to inner leaflet. It contains only two mycolic acid and arabinine. These mycolic acids are linked to the termini of the arabinine chain to form the inner leaflet of a pseudo-lipid bilayer of electron transparent zone of cell wall. Electron transparent zone of cell wall is finished. Now we will move to electron dense zone. Why it is very dense? Because of the presence of peptidoglycans. Peptidoglycans are nothing but chains of alternating N-acetyl glucosamine, N-glycolyl muramic acid linked by peptide bonds, which are again linked to galactan layer by arabinogalactan. Already I told you in electron dense zone it contains arabinine, galactan, lipomannan and peptidoglycan. Here inside the peptidoglycan it contains NAG, N-acetyl glucosamine, NGM, N-glycolyl muramic acid linked by peptide bonds. Coming to cell membrane, it contains proteins responsible for active and passive transport of substances between inside and outside the cell, from outside to inside the cell and from inside to outside the cell, transport of substances, and enzymes responsible for synthesizing cell wall and capsular lipids. These two layers which are present above cell membrane, both cell wall and capsule, is produced from enzymes from cell membrane. It mainly contains two proteins known as major membrane protein 1, which is about 35 kilodaltons, major membrane protein 2, 35 into approximately 10, 380 kilodaltons. MMP1, MMP2. MMP1 is serologically active. It is similar to mycobacterium avium. MMP2 is similar to both avium and mycobacterium tuberculosis. Similar proteins are seen in those organisms also. Coming to cytoplasm. Cytoplasm contains proteins 17 kilodalton protein, 28 kilodalton protein, 65 kilodalton protein known as Grohl heat shock proteins. Now coming from outside to inside, capsule, cell wall, cell membrane, cytoplasm. Inside capsule, mainly two things you have to remember: thiocerol dimycoserosate, phenolic glycolipid 1, 2 P's. Next, below capsule, cell wall. It is divided into two: outer transparent zone, inner dense zone. In transparent zone again divided into two: outer leaflet, inner leaflet. Outer leaflet, two things from capsule: PDM, PGL. Only one extra thing: TMM, trehalose monomycolate. Below it, inner leaflet. It contains only two things: mycolic acid, arabinine chain. And then dense zone. Why it is dense? Because of peptidoglycans: arabinine, galactan, lipomannan. And then innermost cell membrane, two proteins, MMP1, MMP2. And then cytoplasm, three proteins: 17 kilodaltons, 28 kilodaltons, 65 kilodaltons, also known as Grohl heat shock proteins. Now I will draw a diagram and you have to label the parts. Just we will revise with the diagram and we will wrap up. We will draw from inside to outside. First we will draw cell membrane. We'll revise from inside to outside. Innermost cell membrane. It contains MMP1 and 2. Then above cell membrane, cell wall. In cell wall, it is divided into two: electron dense and electron transparent. Why it is electron dense? Because of peptidoglycans. What is peptidoglycan? Alternating N-acetyl glucosamine, N-acetyl glucosamine and N-glycolyl muramic acid, NAG, NGM, NAG, NGM. And remaining three things: arabinine, galactan, lipomannan. Arabinine and galactan known as arabinogalactan. I am drawing now arabinogalactan. What is left over? Lipomannan. This is lipoarabinomannan. It extends from cell membrane to electron dense zone. This is lipoarabinomannan. This is peptidoglycan. This is arabinogalactan. All these together electron dense zone. Then what is above electron dense zone? Electron transparent zone, which contains only two. What I told? Mycolic acid. Now I am drawing mycolic acid. It is attached to arabinogalactan. Arabinine, mycolic acid plus arabinine, nothing but electron transparent zone of cell wall. Now cell wall finished. What is left? Capsule. What it contains? Only two mainly we have to remember: PDM, thioceral dimycoserosate. Thioceral dimycoserosate. Next, PGL1. This is extending to electron transparent zone. PGL1. This is capsule. Electron transparent zone is again divided into inner leaflet and outer leaflet. What is present in outer leaflet? Nothing but capsule structures only. PDM, PGL. Here you can see it is extending into outer leaflet. Only one thing which is extra in outer leaflet: TMM, trehalose monomycolate. This Y-shaped structure what I am drawing is trehalose monomycolate, present in outer leaflet of cell wall. Complicated thing is only cell wall, cell membrane, capsule. Remember the name, the average dermadoc. Like, share and subscribe. Thank you. Hasta la vista. Desvidania.